Hello and welcome to the University of California at San Francisco. In this issue of Cell, the Shah Lab presents its findings on how the brain may control social behaviors. Please join Cindy, Xiaohong, Nirao, and myself as we discuss the findings of our study. Nirao, you're the senior investigator on this study. Can you give me some background about the social behaviors the lab studies?
Males and females of most animals exhibit large differences in many social behaviors. These sex differences or sexual dimorphisms in behaviors can be observed in displays of mating, aggression, and parental care. We are interested in understanding how it is that the brain controls these sexual dimorphisms behavior using mice as a moral organism.
These are complex behaviors. How do you know where to start?
It's been known for a long time that sex hormones secreted by the gonads exert a profound influence on the brain to control sexual dimorphisms in behavior. For example, in males, the testes secrete testosterone, which in turn acts on neurons in the brain to control male typical displays of behavior such as aggression. Similarly, in females, the ovaries secrete estrogen and progesterone, which then acts on the brain to control social behaviors in females. Knowing this fact, we can then ask how is it that sex hormones act on specific neurons in the brain to control sexually dimorphic behaviors.
So Xiaohong, what was your strategy to figure out how these hormones are acting on neurons, and what did you find out?
We know that the receptor for these hormones are expressed in various brain regions, including the hypothalamus. This is very intriguing because hypothalamus is a critical center for mating, aggression, and nursing. So as Nirao mentioned, the hormones are present at different levels between males and females. We reason that the gene regulating hypothalamus should also be differently expressed between males and females. So we therefore compare gene expression profile in adult hypothalamus between the two sexes. What we found is a total of 16 genes that are expressing differently in various hypothalamic regions, as shown in this figure. So the red means it's upregulated in males compared to females, and green means it's upregulated in females compared to males. And the darker the color is, the stronger the sex difference is.
Well, that's pretty exciting. But one, how are the genes differentially expressed? And two, what does it mean for the animal?
As we might have predicted, the sex differences that we see in gene expression are controlled by sex hormones. One example I can give you is CCKAR, which is a gene that is expressed in higher levels in the female hypothalamus as compared to the male. When we remove ovarian hormones, estrogen and progesterone, from the female, we find that CCKAR expression drops to that of male-like levels. When we give back ovarian hormones, we find that CCKAR expression is induced. So what does this mean for the animal? Well, we know that removing sex hormones abolishes behaviors such as mating. Since we also know that sex hormones control the expression of different genes, this suggests that the genes that we've identified, such as CCKAR, may also be controlling different behaviors, such as mating.
In fact, that's what we found. CCKAR mutant females show strong reduction in sexual behaviors. So unlike wild-type females, which allow males to mate with them, these mutants actively reject males' advance, as you can see in this video. So in additional studies, we know that CCKAR act very rapidly to control female sexual behaviors. We know this because when we inject CCKAR antagonists to wild-type control females, now these females start to reject males' mating attempts, just like the mutants.
To summarize then, you find sex differences in gene expression. These differences are controlled by sex hormones, and that the genes control specific aspects of behavior. Is that right?
That's right. As Cindy and Xiaohong have already described, CCKAR is sexually dimorphically expressed in the hypothalamus, and it controls female sexual behavior. In addition, we've tested 3 other genes that also turn out to control specific aspects of mating, fighting, or parental care. So taken together, each of the 4 genes that we've tested so far control specific components of mating, aggression, or parental care in one or the other sex. That's not to say, however, that we are saying that single genes control these complex behaviors. Rather, we believe that our findings suggest that there are complex networks of genes, each of which controls specific aspects of these complex social behaviors. Nevertheless, our findings suggest or provide a genetic framework for understanding how such sexually dimorphic behaviors are controlled.
Thanks for joining us. You can find more information about these exciting results on the Cell website.